All right, so just a couple of quick things before we start. So today we're going over chapter 4, which is a tour of the cell. Um, Today I'm also uploading a quiz to Canvas. Well, actually, sorry, tomorrow, Thursday, I'll be uploading a quiz to Canvas for both sections. You will have till the end of Saturday to complete, okay? So most likely tonight, but you'll technically have till tomorrow morning for the quiz to start, okay? Um, the quiz will be provided on Canvas. Um, should be relatively simple to access. If you go to Canvas, you're on your Canvas shell, you can just go to quizzes and there will be quiz 1 in course quizzes, okay? So it should be fairly simple to get to. It'll be just quiz 1 and then you'll have the quizzes and tests consecutively after that, okay? So there's nothing here. You will not be able to see the tab right now because there's no quizzes. Once I upload that quiz, you'll be able to access this little tab just above the McGraw-Hill Connect tab on your Canvas shell, okay? And that's where you'll access the quiz. It will be timed. You will have 10 questions, and it's 10 points. And yeah, this should be it, okay? So we'll go ahead and get started. All right, so chapter 4, tour of the cell. All right, so cells. So it's important to utilize different types of microscopy as well to visualize cells, right? So this is a light micrograph, or this is just a regular compound light microscope, and this is filming or an image of a eukaryote, okay? And how can you tell this is a eukaryote? Well, one of the main things is that you can see individual organelles. So these are individual pockets of what appears to be vacuoles and nuclei and things like that that are present in this cell, okay? But again, photographs taken with the microscope are called micrographs. Light microscopes can magnify up to 1,000-fold. Electron microscopes use beams of electrons rather than light and can show objects about 100 times smaller than light microscopes. Excuse me. So the only way we're able to visualize our cells or different types of cells is typically through microscopy, okay? And there's different types. We have the light, we have electron, we have different types of light microscopy. We have fluorescent microscopy, two-photon microscopy. We won't get into that, but just know microscopy is used to look at microscopic cells, so forth, okay? Here we have an electron micrograph, and again, you'll have much higher magnification and much higher resolution with an electron microscope, right? So here you see a paramecium, and you see all the different little cilia hanging out on the outside of that cell, and you can see you have a much higher degree of resolution here, okay? The difference between scanning electron microscopy and transmission electron microscopy. Scanning will have the surface being exposed, whereas transmission will have the electrons passing through that specimen here, okay? And you're able to see internal structures as is shown here. You'll see the internal vacuoles, the nucleus, things like that. Also, you'll still be able to see the cilia, which is on the outside, right? But you'll see just a single layer of that cilia rather than all these external appendages kind of coming outside of that cell. All right. So this is just a nice little introduction to microscopy. Okay. You have again light microscopy and scanning or electron microscopy. Okay. And this is just to show you that we need microscopy to visualize the cells. Okay. Organisms and cells. Organisms can be single-celled, such as bacteria and protists, or multicellular, such as mammals and plants, right? So you can have single-cell organisms, right? We should already know this. Bacteria, small eukaryotes, those are all going to be single-cell, right? And we do know that we have multicellular organisms, such as ourselves, your dog, elephants, right? Whales, plants, okay? even fungi, which we'll get into a little bit later when we talk about fungi. So most cells range in size from about one micron to about 100 microns. Okay, so 100 microns again is going to be on the larger side. You'll see mainly the eukaryotes or things with nuclei at this higher range of size. Okay, things at 1 micron are going to be bacteria or smaller eukaryotes. We typically will see bacteria being small. Okay, so many bacteria are much smaller than your cells or let's say an animal cell. Okay, some bacteria can even live inside of your cells. Chlamydia is a bacterial organism that actually can live inside of eukaryotic organisms or animal cells, right? So bacteria can be much smaller than animal cells or eukaryotes, which we'll talk about in a little bit, right? So animal cells mainly is what we're referring to, right? There are some that's a typo. Some exceptions, such as an ostrich egg, which is about 6 inches, and a giant squid nerve cell that can be more than 30 feet long. These are individual cells, okay? So individual cells that are very, very large, and they're very special cells, right? An ostrich egg is just one big cell, and it can weigh up to a pound, right? That big 6-inch egg is technically one single cell. A giant squid nerve cell is just one long nerve cell, okay? And that's very, very unique. That's very different. Most cells, again, are in this 1 to 100 micron size, okay, which is very, very small. You still need a microscope to observe 100 microns cell size, okay? So cell theory, the theory that all living things are composed of cells, and that all cells come from earlier cells, okay? So this is just referring to how life works, right? Cells come from cells, cells make up life, and all cells come from cells, or all life comes from cells, right? So this is essentially referring to cells as the smallest entity of life, okay? Nothing is smaller that's alive than an actual cell, right? And again, we can have very, very small cells such as bacteria, but anything below that, anything smaller than that, you can't have life. Okay, cells are the smallest denominator of life, or smallest fraction of life, right? Okay, so here's kind of a relative size comparison, right? So we have human height, we have the length of some nerve and muscle cells, just close to 1 meter. We have a chicken egg, again, very large cell there. Frog eggs, getting smaller. And here we get into what is able to be visualized by a light microscope. We have a paramecium, which is a larger eukaryotic cell. We have plant and animal cells here, a nucleus, most bacteria, mitochondrion, which we'll talk about later, which is actually remnants of ancient bacteria. Very, very small bacteria like Mycoplasma. Mycoplasma causes walking pneumonia, and again, it can be, it's very, very small. It's hard to even visualize with a microscope. Okay, so this is on the fringes of what light microscopy can do. And then we have the electron microscopy. Again, you have much higher resolution. You're able to see small bacteria, viruses, ribosomes, which are tiny, tiny, tiny proteins, lipids, and then this is where it ends, and you have small molecules, right? And atoms, which we know are the most smallest of anything, right? The smallest common denominator, or smallest common denominator for everything, okay? All right. So the two major categories of cells, I've been kind of throwing those terms out earlier, right? Prokaryotes and eukaryotes. So a prokaryote is a type of cell lacking a nucleus and other membrane-bound organelles. Prokaryotic cells are found only among organisms of the domains bacteria and archaea. Okay, and these are going to be very, very simple organisms when compared to eukaryotes, right? They don't have organelles, they don't have a nucleus, they have free-floating circular DNA and ribosomes and enzymes and cell walls, things like that. But again, they're very simple in comparison to a eukaryotic cell, and we'll see why in a minute. Okay, so eukaryotes are a type of cell that has a membrane enclosed nucleus and other membrane enclosed organelles. Okay, All organisms except bacteria and archaea, including protists, plants, fungi, and animals, are composed of eukaryotic cells. All right. So this is kind of pointing to differentiation in the cells. This is kind of pointing to complexity, right? So, and this is common when you get into higher orders of life, right? Such as humans or developed mammals. You start having separation of jobs in the body, right? You start having separation of compartments in that cell, right? So having these little organelles or having these compartments or vacuoles in that cell provide complexity, right? And this cell must be bigger. It has to be bigger and larger because it's just more complex, right? Compared to the prokaryotic cell. Complex, this cell's incorporated into higher order life, such as humans, right? Our cells are eukaryotic cells. They have different types of organelles. They have different types of functions. You have nerve tissue. You have tissue or cells that make up your different organ systems, right? So you can differentiate these cells much greater in much greater detail than you can the prokaryotes, right? You don't see smaller cells making up larger organisms, like prokaryotes making up larger organisms, such as like a cat or a dog, right? This type of complexity in prokaryotic cells doesn't exist, only in eukaryotes, okay? So here we have the comparison between a prokaryote and eukaryote, right? First of all, approximately 3.5 billion years ago, the first eukaryote evolved 2.1 billion years ago. So prokaryotic cells have over a billion years of extra time here on Earth, right? Showing that this is much more primitive than the eukaryote, right? Again, alluding to the complexity differences between eukaryotes and prokaryotes, right? So prokaryote cells are found in bacteria and archaea. Eukaryotes found in protists, plants, fungi, and animals, right? So prokaryotes, again, smaller, simpler. Eukaryotes are larger, more complex. Again, they have separation of functions inside the cell, which is, alluding to the complexity to that cell. So prokaryotes, most have cell walls. Some have capsules, fimbriae, and or flagella. Again, those are all characteristics of bacteria. And then in eukaryotes, you have plant cells have cell walls. Animal cells are surrounded by an extracellular matrix. They both have plasma membranes, which is important. One has organelles, which is a eukaryote, right? Separation of functions inside that cell. And bacteria or prokaryotes do not, okay? Prokaryotic cells have circular DNA that's not enclosed inside of a nucleus or another envelope, whereas eukaryotes have a nucleus containing one or more linear chromosomes. So that's going to be found inside that nucleus here. So they both have ribosomes, right? So this is actually a really important slide to know. I'll probably ask a lot of questions off of this slide specifically, okay? So just know the differences between the two types of cells that we have. Remember, this is mainly bacteria and archaea, but you mainly can think of prokaryotes as bacterial cells. Archaea are yeah, don't worry about archaea. We'll talk about that later. But just know that this is typically bacteria. This is typically single-celled or multicellular organisms are from the eukaryotic cells. Okay, they give rise to eukaryotic cells. All right. So what do all cells have in common? They have a plasma membrane, which is a thin double layer of lipids and proteins that sets a cell from the surroundings and acts as a selective barrier to the passage of ions and molecules in and out of the cell. Consists of phospholipid bilayer in which proteins are embedded. Oh, that was a mouthful. Okay, so plasma membrane, you can think of it as a selectively permeable membrane. So this is very similar to that dialysis tubing we were using in that online laboratory, right? So it's going to allow for certain molecules to come in, certain molecules to come out. But again, both prokaryotes and eukaryotes have that structure, okay? So cytosol, this is a fluid part of the cytoplasm in which organelles are suspended. So just this juice that's found on the inside of that cell, right? That's right. I guess juice is probably the easiest way to say it. The pulp or the juice that's found inside of that cell that contains proteins, nutrients, things like that that want to be kept inside that cell. All right. So they both have chromosomes. So a gene structure found in the nucleus of a eukaryote cell and at the center of a prokaryotic cell. Okay. So there are bacterial chromosomes. They are circular, but they are not enclosed in a nucleus, right? So this is not a nucleus. There's no envelope here, right? So a bacterial chromosome is one big piece of circular DNA. It's kind of all mushed together in the center of that cell. Eukaryotes are organized very differently. They have chromosomes. They have arrangements or proteins associated with the genetic information, like histones and the packaging of that genomic information. Again, typically eukaryotes have larger genomes, right? They have more genes they need to in order to survive, whereas bacteria can have a much more simplified genome, right? Because they don't have all these complex structures and differentiation within that cell, right? So eukaryotes, by the way, every single cell in your body has the information that makes up you. So from one of those cells, you can essentially grow another human if you have the right conditions, right? Even during cloning, that's what they can do. They can utilize a single cell from, let's say, a sheep or whatever they're trying to clone, and they can essentially take that nucleus out of that cell that they're trying to clone and insert it into a fertilized egg. And you can have the differentiation and then eventual development of that clone, okay? Which is interesting, and it's amazing to think that any one of your cells that you pull off your body has all the information needed to make you again, okay? And that's something very, very special about eukaryotic cells, especially when they're associated with eukaryotes that have multicellular or multicellular organisms, right? Such as you and me. We have all of these cells, and all of these cells have packaged up DNA that has the information to make up you. Which I think is quite impressive, right? Having all that information in every single one of your cells. All right, so ribosomes, a cellular structure consisting of RNA and protein organized into two subunits and functioning as the site of protein synthesis in the cytoplasm, okay? So you do need to know this. This is a good slide to remember, right? What do all cells have in common? Plasma membrane, cytosol, chromosome, and ribosomes, okay? Very, very important. Again, ribosomes will make proteins, okay? So both cells utilize proteins. That makes sense. Proteins are a commonality amongst all life, okay? All right, so eukaryotic cells, they have organelles, a membrane enclosed structure with a specialized function within a eukaryotic cell. And they also have a nucleus, so plural nuclei. Remember, the first definition I gave you is an atom's central core containing protons and neutrons. And then the second definition, I'm giving you the genetic control center of a eukaryotic cell, right? So this is the center of that cell that houses the chromosomes or the genetic component of the eukaryotes, right? And again, this is only found, these two structures, well, organelles or many structures and the nucleus are found only in eukaryotes. All right, the prokaryotic cell shown here. So this is a micrograph of Helicobacter pylori, a bacterium that causes stomach ulcers. It also can cause stomach cancers. It's quite nasty. Is shown alongside a drawing of an idealized prokaryotic cell, right. So yeah, so here you have a simple bacterium, and you have flagellum, and here you have the analogous structure shown here. And again, remember that all the cells have a plasma membrane. They do have the cytosol, they do have a genetic component, and they do have ribosomes. So bacteria have ribosomes free floating in the cytosol. They have this nucleoid, and they can also have differential structures such as flagellum or capsules, cell walls, things like that. Not all bacteria will have these structures, but typically when you see the images in the book, they will have this flagellum, which is for movement. You'll have different coatings on the cell wall or the external cell structures, but the main components that will always be there are the cytosol, cytoplasm, the plasma membrane, and then the genetic components and the ribosomes, okay? Also, bacteria or prokaryotes can have different shapes, right? So one thing you'll note is that you'll see many bacteria with this rod shape. They can be a circular shape, they can be a sickling shape or a vibrio shape, which is like a curved bent rod. You can have a spirochete. There's a lot of different shapes of bacteria or prokaryotes, right? They even have some square and star-shaped bacterial organisms, which is quite interesting, okay? But they're not held to this shape standard, right? Not all prokaryotes are shaped this way. But again, remember that all prokaryotes have that plasma cytosol genetic component ribosomes, okay? All right. And here's a typical animal cell or plant cell, which you would see this is more going to be an animal cell. Again, plant cells will have something, a cell wall, again, to protect themselves from rupturing during osmotic stresses and pressures, which we'll talk about in a bit. So you do need to know these different components. We will go through these individual organelles, but what we have here is the commonalities amongst prokaryotes, right? We have the plasma membrane, you have the cytosol, you have a nucleus which houses that genetic component, and then you have ribosomes. But in eukaryotes, you have ribosomes that are free-floating, or you have ribosomes associated with this structure here called the endoplasmic reticulum. And again, this endoplasmic reticulum is going to be utilized for packaging or processing proteins for secretory purposes or secretion, right? So dumping out into their environment, or just changing or processing those proteins in that organelle structure, okay? And don't worry, we'll get into all these different structures right now, okay? So here we have a plant cell. Again, the major differences that you'll note is this cell wall structure. So remember, we talked about cellulose. It's the exact same sugar that makes up starch and glycogen too, right? So this is a bunch of glucose molecules linked together, right, to form a very organized structure, a very strong structure. And this is that cell wall. Remember, this is fiber that we talked about. Very, very difficult to destroy. You cannot break this down with any enzymes that you have, any digestive enzymes that you have. Again, you need microorganisms or prokaryotes to break this down. We do not make the proper enzymes to break this down. You need either a fungus or some other microorganism that can destroy the cell wall. And the main function of that cell wall, again, is to protect against osmotic stresses. Osmotic stresses or water pressure, right? So plants have different changes in pressures. They pull in water, and they need to have a cell structure that prevents those cells from popping or rupturing under that stress, okay? You also have a central vacuole that can be used for storage, or it can be used for maintaining the turbidity or the turgidity of that cell. So again, this vacuole can be filled with water and provide pressure for that cell to stay upright, right? So plants, if they have sufficient water pressure, they can stand up straight from that water pressure, right? Or if you lack water pressure, you have the plants wilting, okay? So again, very, very important. But you have the same structures that you would have with a eukaryote. The big differences are the vacuole cell wall and this organelle here, which has not been mentioned or even labeled, but this is going to be the chloroplast, okay? And this converts light energy, or this harvests light energy to make, or carbon fixate, right, to make sugar, all right? So plants do have the ability to pull CO2 from their environment and make sugar. But we'll talk about that in a minute. All right. Oh, well, it does have a chloroplast here. Yeah, so not in animal cells, right? So central vacuole, cell wall, and chloroplast. All right, so the plasma membrane. So it's phospholipid, a molecule that is part of the inner bilayer of biological membranes, having a hydrophilic head and a hydrophobic tail, which we'll talk about in a minute. So phospholipid bilayer, a double layer of phospholipid molecules, each molecule consisting of a phosphate group bounded by two fatty acids. That is the primary component of all cellular membranes. So what do we know about fatty acids? If you don't remember, those are going to be those saturated hydrocarbons, right? So carbons bonded to a bunch of hydrogens. Um, One of the main things to note about that is that it's nonpolar, right? So this fatty acid tail are going to be nonpolar. They're going to be resisting water, right? And you do know that that phosphate is going to be hydrophilic, right? They're going to be water loving. So you'll see that here. So again, you have the hydrophilic head, which is that phosphate group, and you have that hydrophobic tail. So this organization of this molecule is extremely important to life, okay? And this is this is common amongst all life. You are required, or the cells are required to have this phospholipid bilayer. Again, these are the phosphate heads that are gonna be attracted to the water. And here we have the hydrophobic tails that are gonna be not attracted to water. They're gonna resist water, okay? So these on the inside of this cell or plasma membrane, this phospholipid bilayer is resistant to water, keeps water out. The heads, again, are going to like water, so they'll be bound or they'll be attracted or flipped over head to tail. Right, so this is the bilayer is referring to each side having one phosphate, and then the fatty acid tail, which is hydrophobic, the hydrophilic head. And you can see that this can separate the inside and outside of the cell very nicely, okay? This is selectively permeable. Water can pass through, small molecules can pass through, but charged molecules cannot pass through. Again, charged molecules will be held up by this phospholipid fatty acid, okay? Right? It can get here, and then the charged molecules will not be allowed in, right? So this is a very, very nice barrier. Again, water can come in and out, but charged molecules cannot. Large molecules cannot. Again, very, very effective barrier at protecting that cell, okay? And this is required for all life, right? This phospholipid bilayer, okay? So here we have a more complex, I guess a more complex image showing the phospholipid bilayer. And remember, this is going to be that layer of that structure here, right? This is going to be that plasma membrane shown here. This is this phospholipid bilayer that's going to be surrounding that cell, okay? So it's important to note that it's totally encompassing that cell, separating the inside and the out, okay? No matter if there's a cell wall present, right? So here is that plasma membrane shown here. Again, this has a cell wall associated with it, so it's an extra layer of protection. But again, keep that in mind. It's a single layer and it's wrapped around the entirety of that cell. All right. So here we have, again, the more complex image showing the cell wall or the plasma membrane. So again, you have those phospholipid heads and the fatty acid tails that are going to be resistant to charge and resistant to water, and those hydrophilic heads. But you also have these proteins embedded within that plasma membrane, right? And this is going to be typical of what you would see in this plasma membrane. You have these proteins here, they're channel proteins or receptors, things like that, that are going to be present on that cell, okay? And they can either help with diffusion of large molecules, active transport, which we'll talk about later. So they can either move or signal things into the cell. But just note that this is referring to the fluid mosaic model of that membrane. So the fluid mosaic model is just referring to the movement and the embedding of the proteins inside of that membrane, right? And if you think of a mosaic or a mosaic artwork, you can see how this model is representative of that, right? Mosaic artwork is made up of little pieces of individual painted either textile or fragments of glass, things like that, right? And they make up this one big image, and it can be beautiful and large, right? But this is the exact same kind of principle here. You have multiple proteins associated with that that plasma membrane, as well as these different phospholipid heads shown here, right? So it's fluid, right? It's always moving, and it's kind of depicting how that membrane works and how that structure is formed, right? So again, these proteins are not just sitting there in the plasma membrane and stuck. They're bouncing around. They're moving. Receptors are moving. Um, these heads can be uh, moving between different areas of that cell or that plasma membrane, right? So nothing is staying put. Everything is in motion constantly as that cell is living and surviving. Okay, so this plasma membrane is constantly in motion, right? And that's important to know that this isn't just staying there. These things are actively moving. Okay. All right. So going into the organelles, the nucleus. So the nucleus, again, it's going to be the area of the cell, the organelle that's going to be housing the genetic information or the chromosomes, right? Um, so yeah, so nuclear envelope, a double membrane, perforated with pores that enclose the nucleus and separates it from the rest of the eukaryotic cell. Okay, so again, this is going to be kind of that outer or inner shell protecting the genetic information, right? Chromatin, the combination of DNA and proteins that constitutes chromosomes, often used to refer to the diffuse, very extended form taken by the chromosomes when a eukaryotic cell is not dividing. Okay, so this is referring to mainly the arrangement or the organization of the DNA, right? When the DNA is not being utilized, it's wrapped up tightly, almost like a spool or a string that's wrapped into a spool. And again, it's just to organize the DNA to better package the DNA inside that cell. So remember, the nucleus is only found in the eukaryotes. Eukaryotes have much more complex genomes, so their genomes are much larger. And if you don't remember, as I mentioned earlier, each cell in your body, or each cell in a eukaryote or complex organism, has all the genetic information needed to make another one of you, right? So there's a lot of information there. So think about the genes that make up the different cells, such as your skin, your eyes, your organ systems, your hair. There's a lot of genes that are required. There's a lot of information. So in order to organize that DNA or that information, we have chromatin, right? So proteins, again, that are going to be bound to that DNA that organize it in a certain way where it doesn't kind of spill out everywhere, right? It needs to fit inside of that cell. So nucleolus, a structure within the nucleus of a eukaryotic cell where ribosomal RNA is made and assembled with proteins to make ribosomal subunits, consist of parts of the chromatin, DNA, RNA transcribed from the DNA, and proteins imported from the cytoplasm, right? So this is going to be an area where we have the ribosomal subunits made. So there's a lot of translation going on, or transcription, and then the creation of the ribosomes there. Okay, so here we have the nucleolus. Again, this is the area where the ribosomes are put together, right? Proteins and RNA are going to be associated. So in order to get a protein back into the cell, it has to leave, get transcribed, and come back in to this this area. Okay, so protein typically doesn't get made here. It has to come outside to a ribosome, which we'll talk about in a minute. So here we have the nuclear envelope with these different pores. So these pores, again, are little cuts in that nucleus, again, where messenger RNA or proteins can come in and out, which is important, right? Chromatin, which is going to be that condensed DNA, and then, yeah, that's pretty much it. Right. We'll talk about nuclear pores and the envelope, I guess, in a minute or late. Yeah. So here we have the nucleus. This is an electron microgram or transmission electron microgram. Again, electrons can have a higher level of resolution, so you cannot see this with a microscope. You need to have electron microscope here. So here you see the surface of that nucleus. And you have these pores. Again, if you look closer, you see these individualized pores here, which are going to have these proteins associated with it. Again, having these pores allow for the exit of mRNA or the entrance of protein. into that nucleus. Again, the only reason, or there's a few reasons why to have protein inside that nucleus, but one of the main reasons is to make ribosomes, right? All right. Oops. So here we have the DNA, chromatin, and the chromosome, right? So again, the DNA molecule is going to be that information or contain the genetic information that can then be used to give that information to make up whatever that organism is, right? If it's bacteria, it'll be a little different. They don't have these chromatin or these histones associated with the DNA, right? So these are individual histone proteins. Histones with DNA will make this chromatin, okay? Again, this is important to package up the chromosome this way, again, to organize the information because there's just so much, okay? You can't have the free-floating DNA like bacteria have because bacteria, again, are much simpler, or prokaryotes are much simpler. You need to package up the DNA in eukaryotes into the chromosome to organize it inside of that nucleus. Everything needs to fit there, right? So very, very difficult to do. And if there's so much information there that it can be difficult, right? Yeah. So again, just know that you have DNA as a storage molecule for the genetic information of that cell. They can make up all of that organism, right? All this information is housed inside of one single cell. You do have these histone proteins, or again, chromatin is going to be histones mixed with DNA, and then it packages up that DNA into a nice chromosome. Therefore, it can fit inside of that nucleus, okay? So it's essentially what this image is referring to, that organization of the DNA inside of a eukaryote. All right. So ribosomes. So ribosomes inside of a eukaryotic cell. Again, we're mainly going through eukaryotic cells right now, as bacteria or prokaryotes are fairly simple. Here you have free-floating ribosomes, and you also have ribosomes associated with the endoplasmic reticulum. But ribosomes, their job, or what this organelle is responsible for, is making proteins. Okay, this is a very, very important organelle. Again, the nucleolus is where ribosomes are made or created, right? So what that means is that you need ribosomes in order to make ribosomes, which is kind of funny, right? Ribosomes are made up of both RNA and protein. Okay, so you have a ribosome processing an mRNA to make protein. We will get into this this mechanism later on. We talk about genetics, but essentially what you have is, right, DNA to RNA to protein. This RNA is going to be produced or created in the nucleus, and then it's going to leave one of those nuclear pores out of that envelope, and then go to a ribosome, where it then can plug in and make the protein. Okay, and again, we'll get into this this process or this mechanism a little bit later. This is really complex, and we probably won't get into that much detail with this right now. But again, just know that this is mRNA. Again, mRNA originates from the nucleus, plugs into the ribosome, makes the protein. Okay. So here we have the ribosomes attached to the endoplasmic reticulum, visible as a tiny dark blue dots. So we'll talk about the endoplasmic reticulum in a minute. And it's kind of important to have this rough endoplasmic reticulum, especially closely associated with the nuclear envelope. Again, ribosomes' main job is to make proteins, okay? So inherited genes in the nucleus control protein production and hence the activities of the cell, right? So again, you have the central dogma here. You have the DNA into RNA into proteins, right? So again, DNA can be just transcribed into mRNA. mRNA can then leave that nucleus through a nuclear pore, and then it can plug into a ribosome, wherever that ribosome may be. Okay. After that, it can synthesize a protein through the use of that ribosome, right? And these are going to be, again, amino acids, right? We talked about amino acids, the monomer to proteins, right? The individual amino acids are linked by the amino group and the carboxylic acid group. again, making up this large molecule. Again, DNA, RNA, protein, mRNA needs to leave to plug into the ribosome and the cytosol, whether it's in the endoplasmic reticulum or in a free-floating ribosome. Okay? So again, ribosomes make proteins. So the endomembrane system, manufacturing and distributing cellular products. So a vesicle, a membranous sac in the cytoplasm of a eukaryotic cell. Endomembrane system, a network of organelles that partitions the cytoplasm of eukaryotic cells into functional compartments. Some of the organelles are structurally connected to each other, whereas others are structurally separate but functionally connected by traffic of vesicles among them. That is a mouthful, okay? So just know that the vesicles or the endomembrane system are essentially a packaging or processing plant of the cell, okay? And there's a lot of similarities, a lot of overlap between like the Golgi and the endoplasmic reticulum. The endoplasmic reticulum essentially makes certain types of secretory molecules or things that you can secrete. One thing I think of is like mucus, right? So your cells can produce mucus, especially in your upper respiratory tract, right? Those are glycoproteins. And glycoproteins are sugars mixed with proteins. So, and essentially what your mucus does, or what it's utilized for, is you have all this snot, right? Especially, it's annoying when you're sick, but it has a purpose. So you breathe in and out, you can trap microorganisms in there, but the snot or the mucus has to come from somewhere, right? And it's through this endomembrane system where we have the packaging and the production of this mucus, right? You have one glycoprotein or sugar protein that's going to be made in that cell and then dumped out into the environment, so it can help protect you from microorganisms that may infect you. Okay, so endomembrane system, very important for the cell. A lot of biological functions are responsible for are made possible by this endomembrane system, rather. Okay, so very important, right? So endoplasmic reticulum is a part of that endomembrane system. So you have the rough endoplasmic reticulum here, and you have the smooth endoplasmic reticulum here. The differences between the two are the rough endoplasmic reticulum has the ribosomes associated with it. They look like they have little tiny balls kind of sprinkled throughout that ER or the endoplasmic reticulum. The smooth is just not going to have ribosomes, right? And remember, ribosomes make proteins, okay? So endoplasmic reticulum, an extensive membranous network, and the eukaryotic cell continuous with the outer nuclear membrane and composed of ribosome-studded and ribosome-free smooth regions, right? So the rough ER, or the rough endoplasmic reticulum, is a network of interconnected membranous sacs in a eukaryotic cell's cytoplasm. Rough ER membranes are studded with ribosomes that make membrane proteins and secretory proteins. The rough ER constructs membrane from phospholipids and proteins, which is very important, right? So the rough ER is responsible for making membrane and proteins, right? So membrane and proteins for the membrane, okay? So another thing too to note, again, that rough ER, that rough endoplasmic reticulum, is closely associated with the nuclear envelope, okay? So why is that important? Well, it's really important because the rough ER also has the ribosomes associated with it too, okay? So think about that. The mRNA is being made here inside the nucleus. The mRNA can then leave directly to a ribosome, or that is associated with the endoplasmic reticulum. That ribosome can make the protein and then dump it into this endomembrane system where it can be packaged and sent or secreted, or changed in some way in the endoplasmic reticulum. A sugar can be added there. Different other proteins can be associated here and put together. So this makes sense why this packaging site is very, very close to the nucleus, right? Especially the nucleus is dumping or making RNA in order to make these different proteins and package up these different proteins. Okay, so very important. All right. How the rough ER manufactures and packages secretory proteins, right? So you have one, a ribosome, again, makes the protein or the polypeptide. again, by adding multiple amino acids together in this chain formation. This can be then dumped into this endomembrane system or the endoplasmic reticulum, right? So this protein can then be modified, changed. A sugar can be added, right? So I talked about mucus or snot. That protein will then have a sugar added. Let's say this is mucus. A vesicle will then be made where the protein can then be transported out. The vesicles can bud off from the endoplasmic reticulum. Proteins are secreted from the cell when vesicles fuse to the plasma membrane, and then it can be dumped into the environment that way, okay? So a lot of different secretory proteins or secretory molecules are packaged this way, right? And it's important, right? You have mucosal surfaces are going to be the main example that I use because those cells are actively producing a lot, but it can be anything, right? It can be anything in any biological system that's being secreted or dumped into the environment, okay? So immunoglobulin or other types of secretions that your body has will dump those proteins off that way, okay? So very important. So the smooth endoplasmic reticulum, a network of interconnected membranous tubules in a eukaryotic cell cytoplasm. Smooth endoplasmic reticulum lacks ribosomes. That's what makes smooth ribosomes different than rough, right? The rough is referring to the ribosomes. Enzymes embedded in the smooth endoplasmic reticulum function in the synthesis of certain kinds of molecules such as lipids, right? So liver cells, again, important to produce certain types of lipids. You also, in your gametes or your sex cells, you produce sex hormones, right? So your testes or ovaries, you have a lot of smooth endoplasmic reticulum because, again, you make either estrogen, progesterone in females, or males will have estrogen, progesterone, or estrogen as well. And then males will make testosterone in their testes, okay? So again, very important, smooth endoplasmic reticulum, mainly lipid or fat-based secretions, right? So important there. So Golgi apparatus, an organelle, and a eukaryotic cells consisting of sacs of membranous, sacs that modify, stacks of membranous sacs. Wow, that rhymes, okay? So an organelle and a eukaryotic cells consisting of stacks of membranous sacs. Oh, it's kind of funny. What you'll see too is this Golgi will kind of have like layers to it, and you'll see it right now. So these sacs that modify, store, and ship products of the endoplasmic reticulum. And again, it's just another a component to that secretory system, right? This can package up, bud off different secretory proteins or molecules out into the environment or out into your tissues, okay? So here you have the Golgi. Again, this is referring to the stacks. There's different membranes. So this is receiving side of the Golgi apparatus. So you'll have the vesicles come here, they'll fuse, and then they'll be processed, and then new vesicles will form and bud off into where they need to go. So it can be some other area of the cell. It can be out into the environment, outside of the cell. It just depends on what that molecule is, right? The Golgi apparatus consists of flattened sacs arranged something like a stack of pita bread. Oh, that's a typo. That's a book typo. The number of stacks in a cell from a few hundred correlates with how active the cell is in secreting proteins, okay? So again, this is very, very heavily dependent on what that cell type is doing, right? This can be something in the upper respiratory tract where it can secrete various proteins or molecules to protect that upper respiratory tract. It can be in your gut or your stomach, secreting mucus to protect it from the stomach acid. Again, it just depends on the cell type, okay? So here you have the receiving end of the Golgi. Again, the Golgi will have the processing done, right? So the transport vesicle fuses, and then you have the eventual transformation or changing of whatever was in that transport vesicle. Transport vesicle from the Golgi apparatus can then fuse to the plasma membrane and then dump out whatever contents it has into the environment, or it can move that vesicle elsewhere in that cell, okay? All right, so lysosomes. So here we have lysosomes kind of spotted here. Again, these are going to be those vesicles that are free floating in the cell. And they have two main functions. So digestion of food or breakdown of molecules or damaged organelles, okay? And this is kind of important for the cell. So cells can have damaged cellular components, and it's important to be able to break those cellular components down. Um, there's also there's also immunological importance to these vesicles. Cancer cells can be targeted by your immune system to cease or to stop growing and to process itself in something called apoptosis. What that means is that your immune cells or your immune will go and target that cancer cell and say, hey, target cell, or please, you need to die, you're cancerous. And these lysosomes have a part in that, right? They're going to help break down that cell and start the self-destruction process, okay? So again, digesting food molecules, we are heterotrophs, we are catabolic, we need to break down food molecules, right? So this lysosome or the digestive enzyme can then break down various sugars, various food components, lipids, fatty acids, things like that, okay? And lysosome will fuse to a food vacuole where it can break it down. This is a very important process in single-celled organisms, right? So something like a paramecium or an amoeba will require lysosomes to break down their food molecules because they're actively out in the environment. Whereas we're pretty spoiled. We have a digestive system, so we can get glucose to various parts of our cells relatively quickly without too much processing, okay? So again, lysosomes can also break down damaged or dying organelles or itself, right? It can have something called apoptosis where self-destruction occurs, right? And this is typical of cancer cells. And again, it's activated by your own immune system to break down that cell, okay? But for all intents and purposes, just know that it can process or break down organelles that are faulty, or it can process food vacuoles, okay? Break down that food. Yeah, so I like to think of this as in the second image here, the breaking down of molecules or damaged organelles. You know, think of a city, right? Your cell's a city. You have multiple buildings. One building becomes uninhabitable or not able to be utilized anymore. You demolish that building, right, to save the resources of that area. That's essentially what you're doing here. You're just demolishing that that organelle that's no longer serving a purpose for that cell, okay? All right, so vesicles, this is showing vesicles containing damaged organelles, organelle fragments, and those digestive enzymes are breaking down those organelles, right? So you'll see little bits and pieces of those organelles being destroyed, okay? So vacuoles. So vacuoles are very important in plant cells. Again, storage, secretion, it can house poisons or toxins. Caffeine is considered like a toxin that prevents certain types of plants from eating that, or certain types of organisms from eating that plant, right? So you also have contractile vacuole. So contractile vacuoles are found in paramecium. So a contractile vacuole fills with water and then contracts to pump the water out of the cell. This can be utilized for movement as well as regulating osmotic stresses, okay? And we'll get into osmotic stresses later, hypertonic, hypotonic, and isotonic. But this essentially can be used for movement as well as dumping water out. So think of this as a way the cell can move as well as a way a cell can essentially urinate, right? It can, regulate how much water is inside of that cell, right? You can dump out or pull in, okay? So central vacuole in a plant cell, again, a central vacuole colored in blue in this micrograph is often the largest organelle in a mature plant cell. Again, this is going to contribute to the how rigid that plant is and how well that plant is able to stay upright, especially if it's not like a tree, doesn't have that dead or phloem and xylem that are really sturdy, so or wood associated with it, right? So these are going to be like those flowering plants, small plants. They require a central vacuole in order to maintain how stiff those plants are because of that water pressure, okay? So this is also why plant cells have that cell wall, okay? The cell wall prevents the cell from popping. You cannot have this in an animal cell, right? If you soak an animal cell in water, 100% water, that cell can pop. And this is why if you ever go to the hospital, they give you 0.9% saline solution, right? Um, and you're like, what saline solution? Why would they use saline solution? Because your cells have an osmotic range, right? If you pump pure water into a body, um, you essentially can rupture cells in your body because of the amount of water that will rush in. Okay, you need to have some salt to have that balance, and we'll talk about saline and we'll talk about osmotic stresses later on. But just know this vacuole again will contribute to the plant cells and keeping that tree or that plant upright without wilting. Right, that plant doesn't have water; it wilts. This water, again, associated with that central vacuole, will allow that plant to stand straight up. Okay. Also, this vacuole can be used for storage. Plants can make certain toxins. Toxins will be housed here. Various other components as well, storage, things like that, sugars. So chloroplasts. Again, this is going to be specific to plant cells. Chloroplasts are, which we'll get into later when we talk about photosynthesis, but these are the photosynthetic sites, okay? So these are going to be sites where energy is harvested from the sun and then used to make sugars or carbon fixate. So carbon fixation, I'm referring to fixing carbon molecules together, right? So think of pulling in CO2 from the environment, grabbing other CO2, putting it together, and eventually making glucose, right? So C6H12O6, okay? And that is done with in unison with the chloroplast, okay? So again, an organelle found in plants and photosynthetic protists enclosed by two membranes. A chloroplast absorbs sunlight and uses it to power the synthesis of organic food molecules, so sugar. Okay, I don't think we're gonna get through everything. So chloroplast site of photosynthesis. Here we have a thylakoid membrane. Again, these are gonna be those membranes that have pigments that get excited when the sun hits those membranes. Again, very efficient. Very elegant. We'll talk about the specifics and the biochemistry associated with this. But just know plants have these photosynthetic organelles that, again, use sunlight to carbon fixate, use sunlight to make sugar, okay? That's pretty much what you need to know here. That's it. We'll get into the nitty gritties later on. But yeah, plant cells, sunlight to sugar, all you need to know. Okay, and be able to identify it. Mitochondrion, this is also important. These are also in both plant and animal cells. Mitochondrion are the powerhouse of the cell, which everyone kind of refers to, right? It makes ATP. You have an inner and outer membrane. This is going to have a electrochemical gradient, which is, I know it's a big, scary set of words there, but essentially what you have is potential energy, okay? So this is just like, think of a dam, how a dam works. And when we get into respiration, cellular respiration, I'll talk a lot more about the potential energy associated here. But again, a mitochondrion are used for cellular respiration in order to make energy or ATP for the cell, okay? And again, we'll talk about this a bit later. What time do we end? I think we're at 9. Is it 9:25 or 9:10? So maintaining the cell shape. So here we have microtubules or cellular structures that are going to be maintaining how that cell is kept structurally sound. Okay, so these are different proteins that are going to, again, keep that cell appropriately shaped. So microtubules, the thickest of the three main kinds of fibers making up the cytoskeleton of a eukaryotic cell, a straight hollow tube made of globular proteins called tubulins. So microtubules from the basis of the structure, form the basis of the structure and movement of cilia and flagella. So form right here, sorry, there's a typo, okay? So again, microtubules are very, very important. Here you have the cytoskeleton shown of the cell. So this is a micrograph of an animal cell. The cytoskeleton microtubules are labeled with fluorescent yellow dye. So all of these yellow bits and pieces, these squigglies you see here, are microtubules, again, making sure that structure of that cell is kept, right? The structural components of these proteins can also be used to transport different vesicles or organelles around the cell, attachment, adhesion, right? So cells sticking to other cells. Again, very, very important. Okay. Microtubules and movement. The crawling movement of an amoeba is due to the rapid degradation and rebuilding of microtubules. So those microtubules can build or expand, elongate or shorten. And again, this is going to give rise to certain movement strategies from the amoeba. Your white blood cells also move in this mechanism here, right? They can extend or shorten those microtubules in order to get around different parts of your body or cells, okay? Flagellum or flagellin, it makes up the flagellum for sperm cells. Again, these are flagellated, they do move. So what I need you to know here is flagella is different from cilia in that flagellum spins in a corkscrew motion and it has directionality, okay? So it spins like a propeller and it moves in one direction, okay? And that's gonna be a flagellum, okay? So so undulates in a whip-like motion, driving a cell such as a sperm cell through its fluid environment. So again, moving quickly, spinning. Think of a propeller. So cilia, it's a little bit different. Again, utilizing the cellular structures, microtubules and other cellular structures for the anchoring of these cilia. And cilia is a little bit different in that it contracts one way, right? So it pushes it and then it relaxes, pushes and it relaxes. So it moves, it has directionality, but instead of a direction like a whip or like a propeller, it's mainly used for pushing and movement of fluids, right? So think of like a rowboat, right? You're rowing in one direction and you lift up and relax and then you push again, okay? And that's exactly what cilia does, right? So it pushes and then it relaxes, pushes and it relaxes. So a really good example is in the tissues in your upper respiratory tract, the pseudostratified, PCCE, yeah. So pseudostratified columnar epithelium. So that's going to be the lining of your trachea and your upper respiratory tract. And what this does is this moves the mucus up from your lungs out into your mouth or your upper respiratory tract, where either you sneeze it or swallow it into your stomach. I know that's gross, but that's essentially what it's used for, right? You have a very low pH in your stomach that can destroy pretty much anything that you swallow with your mucus, okay? So again, it clears the lungs of debris. Think of this escalator. It's very, very important, again, to make sure your lungs are sterile, right? You have all this mucus lining your upper respiratory tract and your lower respiratory tract. And the cilia movement of that mucus up is a cleansing or pushing any microorganisms you breathe in, any particulates that you breathe in into your lungs up and out, where you can swallow or spit out that those organisms, okay, or those contaminants, right? And again, the cilia is what does that, right? Pushes and then stops, pushes, relaxes, again, pushing that mucus up into the upper respiratory tract. Very, very important. Okay, and that's it for today. Any questions? Any questions before I stop? No? Okay. Well, I'll see everyone at 9:45 if you decide to show up. Okay?